Hi, this is Brian Kavanaugh, and along with my co-host Kaylee Doak, I welcome you to a special presentation of the Virtual Visiting Professor Network entitled A Midsummer Night's Stream. This episode is an exciting live broadcast of the podcast Plenary Session hosted by Dr. Vinay Prasad. Dr. Prasad's guest tonight would be Dr. Sue Yom. Most of you know Dr. Prasad from his academic work and social media presence. He's also the author of two books that should be required reading for anyone interested in medicine. The first is Ending Medical Reversal, co-authored with Dr. Adam Sifu, and the second is Malignant, How Bad Policy and Bad Evidence Harm People with Cancer. Dr. Yom needs no introduction to anyone in the field of radiation oncology, and you'll hear more about her background during the podcast anyway. I'll just show you this beautiful picture of her greeting one of her patients in the lobby at UCSF. I certainly hope that pretty soon we can all say hi to our patients this way once the pandemic is behind us. And so, without any further ado about nothing, we turn it over to Dr. Prasad for a Midsummer Night's stream of Plenary Session.
Welcome, everybody. You're listening to a Midsummer Night Stream version of Plenary Session. I'm Dr. Vinay Prasad. I'm Associate Professor at the University of California, San Francisco. And it's my pleasure to be doing this live, the first ever live Plenary Session recording. And I'm joined by the great Dr. Sue Yom. Dr. Yom is professor at the University of California, San Francisco. She's a practicing radiation oncologist. And many of you here will know her for her superb work in head and neck cancer and in clinical trials. And so I'm going to turn it over to Dr. Yom, who's going to be our guest host for this evening, who's going to coordinate the discussion. But hopefully, we're going to get to learn a lot more about what makes Dr. Yom tick. So thank you so much for being here and joining us in this live plenary session. Dr. Yom.
So Dr. Prasad has invited me / introduced me, and we're going to work it out. So one of the things that I think kind of is really interesting about the two of us being here together, I would say, is that maybe you had a very traditional training and pursued a very interesting and kind of unconventional career pathway, whereas I had very unconventional training and background and pursued a more conventional training. And I've actually been thinking about that a lot this week. Like how does that happen? How does a personal reversal happen as opposed to a medical reversal? So I'm a little curious about that, actually, Vinay. You want to tell me about a little bit, or Dr. Prasad? Can I call you Vinay? I'm going to call you Vinay.
No, please. I invite you to call me by first name, please.
Yeah. How does that happen for you? Or how did that happen?
Well, okay. That's a good thing though to start, and then we'll turn it over to you, and you can take us through, the sort of training that you had and that led you to medicine. But I guess in my case, I did my undergraduate at Michigan State University, which wasn't too far from where I went to high school and where I grew up. My undergraduate was, I took a number of courses in philosophy, and I ended up majoring in that. But I also did all the pre-medical curricula stuff. I think I went into college, I wasn't sure I wanted to go to medical school, but I knew I liked science. And along the way, I got kind of roped into taking some philosophy classes. And then, I applied traditionally right out of college, went to medical school at the University of Chicago. And there, met a number of people who were really sort of stellar people in evidence-based medicine. And I got a really good background in that. And to be honest with you, if I really went back and sort of put myself in the mind of that person back then, I would say, that was somebody who really thought that, I'm just gonna have a private practice career. I'm gonna see patients, and I'm not a lab person. I'm not really interested in clinical trials. And that's gonna be my thing. I'm just gonna be a practitioner, which I continue to believe is, the most noble thing that we do, even if we do other stuff. And then when I was a resident, I think I was increasingly pulled in sort of a research direction when, it was really clinical situations that I encountered. We were in the CCU and somebody put a stent in, and I started to ask myself, why is this patient getting a stent? And I looked into the literature, and I read Courage and Oat, and I read, the clinical trials of that day back then. And I started to have questions about the evidence base. And so, with one of my medical school professor mentors, Adam Sifu, we started to write a few papers. And after a while, you kind of get known for writing papers about medical evidence. It kind of snowballed. And so then when I went to do my fellowship in Hem-Onc, which again, I pursued mostly for clinical reasons, I had this undercurrent of doing research in evidence-based medicine. And at the NCI, I had the opportunity to go over to FDA a little bit to learn from people who did a lot of drug approval about clinical trial design and things like that. And those two things, evidence and trials, they fit nicely together. And so by the time I finished my fellowship, I had done a number of projects and papers in this kind of unique space of maybe you'd call it meta research or health policy. And then when I applied for faculty, which we could talk about, it just kind of continued from there. And so, I think you're accurate in the sense that my current career is non-traditional because I see patients, a couple days in clinic and I attend on service, but my research is not clinical trials. It's not lab. It's health policy, and it's a unique type of health policy that doesn't really fit in many buckets. And so, that's how I found myself doing it. It was never sort of sought out. It wasn't intentional. It was just kind of, I fell into it. Now I wonder, let me turn it over to you.
I find that so interesting because,
okay.
Your story really resonates with me because it just speaks to how much, one single person can really influence the rest of your life. And I've seen that happen. And so, and it's funny because as you get to a certain level, you start to realize that you also have that influence on other people, as I think you probably do now in your current sort of trajectory. And, other people start to as they get further along in their career. And so my story is kind of similar. I was, I was bumping along in, really as a writer and thinking I was, my career was sort of heading that direction with a lot of interest in that. And, some people on the call may not know I did a concurrent PhD in English while I was at the University of Pennsylvania. And I really was thinking a lot about am I going to do psychiatry or medical editing, or am I possibly going to become a journalist? And, Eli Gladstein and Steve Hahn, to a lesser extent, actually just literally changed the course of my whole life. And so it's fascinating to me, you sort of meet this one person and it's that one person who sort of just opens this other complete, really trajectory for you, right?
Yeah.
Yeah.
I guess I continue to think of Adam as, somebody who's both been a mentor to me, but also a friend, a colleague, a collaborator, and somebody who I like a great deal. And he did probably, was willing to work with me on stuff that maybe was even initially, maybe even outside of his comfort zone too, but something that he was happy to kind of work out with me. And that kind of led to, my current career path.
Yeah. One of the things I think about, not to interrupt you, but
No, go on.
there's so much emphasis right now on mentorship and sponsorship. And I'm just going to throw this out there because this is, we've been having random conversations up this whole week, and this is going to be another one. But, I kind of wonder about the magic of mentorship and sponsorship and how that happens, and whether you can really sort of program something like that or legislate something like that. There's all these programs spring up, right? It's kind of like the ice cream du jour, diversity ice cream, mentorship ice cream. But I struggle with that a lot, because how do you make these things actually effective, right? There's a certain match of personality, interests, a certain friction of approach that has to happen for a good mentorship relationship. I don't know if have you mentored anybody? Or have you thought about that? I'm just curious.
That's funny you say that. I cringe when I hear those terms because I feel like I don't always have a good sense of what they mean. And I guess, if you ask me that question, I don't know. Here's what I do know. Whenever somebody who is in training comes to me looking for a project, I think 99 out of 100 times I find something for them to work on. So I've worked with so many people on many, many publications and that sort of thing. But is that really mentorship? That might be part of it. And then the next component is anytime somebody comes to me and they ask me about, should I go into pediatrics or go into internal medicine? Should I go into hem-onc or should I go into orthopedic surgery? I'm always willing to do what I think the best people did for me, which was, I'm happy to get a cup of coffee with you and let's talk about it. And that to me is really a type of mentorship. Whenever people come to me asking about, hem-onc fellows thinking about their job, I'm happy to talk with them about, going to work for Kaiser, going to work for private practice and what that looks like, and let them know experiences of my classmates. But I think many people think mentorship to be sort of apprenticeship. Like, I'm a clinical trialist who does GU oncology, and I'm going to mentor somebody to be a trialist who does GU oncology. And I guess by that definition, I probably don't feel as if I've mentored anyone, because I haven't created anyone who has been so foolish enough to pursue what I have pursued, which is kind of this health policy space.
Yeah, I mean, you are so broad in your interests. I really like that article you wrote in, I think it was Medscape, about the CV wars. And that, obviously that resonated with me. Maybe you want to explain that a little bit, but I can just say, as someone who had a very unusual background coming in, you do sort of wonder, and I am getting to a point with this, which I will in a couple seconds, but you want to explain what you meant by CV wars?
I guess what I meant by CV wars is, I think back about when I was a high school kid. What did I do? I worked as a bagger in Kroger, and I hung out with my friends. And, maybe I did some stuff that we don't talk about on this podcast. But, I wasn't thinking about medical school. I wasn't working in someone's lab. I didn't have any publications. I don't think I published an article until I was a resident. And people of my generation, I'm not that, that far along, but that was considered precocious. That was early to publish an article. A few years before, junior faculty were kind of publishing their first articles. Recently, I got an email from a kid who said, and I'm not using the word kid pejoratively, because this person said, I'm a sophomore. I want to work in your group. And it was a sophomore in high school. And I was like, wow. And the more I look at applications, I see people, doing really things that we used to not see in junior faculty being done in high school students. And so I call this the arms race. And it's a product of the way in which we have created, I think, selection filters and pressures for picking people for medical school. But this is the part that probably you're going to connect about, which is that I believe there is a value to squandering some time in your youth. It helps you figure out who you are and what you stand for.
Does it also make you think more independently? Because one of the things I wanted to get to with this was actually bioplausibility, believe it or not.
Believe it or not. You were going there.
I was going there. Yeah, no, because one of the things I thought was really interesting in your book, and which I thought about like almost the whole time I was reading it, was, why do we love bioplausibility? Like, why is that such an appealing model and concept? And are we like training people to do that? Are we picking people who are sort of inclined in that direction? So bioplausibility, I'm just going to tell you, my view of it. Yeah. It's kind of this idea that you have like this measurement culture, right? You extrapolate these measurements and you feel like you've done an experiment, whether that's on rats or on people. And then you take this measurement and then you sort of translate it into this effect, right? And it's a very experimental sort of approach. And because you have that approach, then you think it models everything else, like everything follows from that model. And, of course, here I'm going to talk about something that radiation oncologists are completely jealous of. Every time I see I'm just going to say it out loud here in public. Every time I see the phrase response rate, it just drives me nuts.
Yeah.
And I can tell you that I don't know how many people in the room share this feeling, but I'm very jealous of response rate. And I'm also very irritated by it because it's not something that radiation oncologists can do. If you've been in radiation oncology for some time, you've realized that we don't actually have response rates most of the time. Most of the time we radiate something and the scan looks, maybe a little bit better. I mean, there are situations where you get a CR, but most of the time it looks a little better. It's a very slow response. You might get a lot of scar tissue, a lot of inflammation. Response rate is not something that radiation oncologists think of. And so if you look at the radiation oncology literature, actually, a lot of it is really based on survivals. And we can talk about radiation oncology literature and how that's evolved from essentially billing records, but that's a whole nother discussion. Basically looking at, survival, because that's what we get. And so I'm curious, maybe you can tell us a little bit about, one of the things I thought was really fascinating was your story of how response rate came about. And why do medical oncologists like that so much? What's up with that?
Oh, because it's things that can be measured become virtuous in and of themselves. But I guess, you got two parts to this question, which I think are really fascinating. So, I'll give you my 2 cents on the two parts. One, why are we seduced by bioplausibility? I guess I would say in part because I think we don't do a terrific job of teaching science. When you, at least I'll speak to my own education, my science classes, even through early college, were not really science like, why do we believe certain things based on what experiments? They were really learning kind of canonical models that were based on many experiments without always understanding the underlying experiments that went into those models. So we think of science as sort of model building, models that people are widely accepted. We don't really understand what were the studies that showed why this model is true and why alternative models dissipated. The second thing I think fits in there is when you read medical history, it is written as sort of an inexorable truth that, why do we give Gleevec in CML? Because, it saves lives. It's a transformative drug. Of course it is. And it has this oncogene. And this oncogene was detected by, Janet Rowley figured out the Philadelphia chromosome. And before that, they knew that the chromosome will break. And, and you look at Brian's early paper in 1996, I think Nature Medicine. And when you give this to cell culture, you can kill the CML cells. It appears addicted to BCR-ABL oncogene. It's a very parsimonious story. And when you look at medical history, you're constantly given parsimonious stories for everything we do. And what you don't get are the stories of all the people, tens of thousands of people who had a parsimonious story, but then it fell apart on phase 3 testing, or it had unintended toxicity. And so I think bioplausibility is in part a way in which human beings write history, because we write to tell a linear story, and we start with what we do and we go backwards. And so it's natural that that's seductive. But I think the important thing that you learn with evidence-based medicine or empiricism is that a lot of this stuff, doesn't really work. There's a huge failure rate, and not all bioplausible things fail. And plausible things and successful things and things that failed were once both bioplausible. I guess the next part of your question, I think, is response rate, which is, a terrific story.
It's an amazing story. I couldn't believe the story. I read the story like 5 times. Like, I was like, how come you didn't write about this for 3 more pages? Yeah, go ahead.
Oh, that's what you
it's like, I wanted to see their faces and stuff while they were doing this.
I know. It's
a good section.
That's the type you need a good writer to write that. But no, I just write the technical drudgery, which is, there's a very famous oncologist, Charles Mortel, in Mayo Clinic. And in the 1970s, he realized something, which was, we give all these drugs to cancer patients, and we need to figure out which of these drugs do we really go forward with in testing? Which of the drugs do we pursue in randomized studies to test what works and what doesn't work? Which drugs are we willing to say that doesn't work? That's ineffective. And he realized that, how do you figure that out in an era before, you couldn't just run randomized trials on every compound? And he realized that, you have to go based on tumor shrinkage. That tumor shrinkage is a marker of activity. Of the millions of candidate compounds in the universe, very few of them will actually shrink tumors. And there's this whole story about Latro, which I didn't talk about, but I'm happy to talk about. But Charles Motel, one evening, he decided, how do you know how much tumors shrink before we can reliably detect that they have shrunk? It's not really it's a question about measurement. It's a measurement question. And he gathered together 16 experienced oncologists. They were, unfortunately, all men. This was the era of the day. They brought the tool that they employed in his clinical practice, which was a caliper or some other tool, and they put a bunch of marbles on a table, and he rolled out foam rubber, and it came in a couple thicknesses, and he had them go around and measure the marbles. But unbeknownst to the audience, 2 of the marbles, or actually 4 of the marbles, 2 and 2, were the same size. And so now Mortel has a really wonderful experiment where he knows how often do Sue and I measure the same marble the same way, and how often and how big and how much different in size do marbles have to be before you and I will reliably agree that they have shrunk. And the answer was 50% bidimensional, which is the WHO indice. The answer was, that's where response rate came from. It came from a study that asked, in an age of calipers, what were the tumor dimensions we could reliably tell apart? And that has been carried forward and eventually morphed into a measure of efficacy. Now you go to ASCO meetings and they say, we got a new drug, it's got a 22% response rate. That's terrific. But that was never what Mortel meant. Mortel meant to use response rate as a way to screen drugs from the many things that were destined to fail. So I think, knowing a little bit about where response rate comes from reminds one that it is not a measure of efficacy in and of itself.
Yeah, that speaks so much to me because recently I got dragged into a whole bunch of AI work. And, one of the things you talk about in AI is called dimensionality, right? And there's this thing called sort of low dimensionality or surface dimensionality, which is that the pattern that you can see on the surface may not actually reflect what's going on. So your story sounds so much like, here's the guy touching the elephant, or here's the surface dimensionality. But what does that really reflect? That whole system is based on what can be measured. Right? It's a self-contained system that has no external referentiality. And then, the other thing, I don't know if you want to say anything about that, but, it just makes me wonder, like, the whole system is so much based on measurement. And, what's it just talks about the fact that response rate really doesn't reflect so much deeper about things, right? So as radiation oncologist, I can tell you, I've run trials. where we used response rate. I had to use response rate. I was forced by the company to use response rate. Okay, full confessions. And I was frustrated by it because, for instance, in skin cancers, response rate is a very nebulous, maybe unusable concept. For three-dimensionality, which radiation oncologists think about all the time, or sinuous structures, which head and neck oncologists think about all the time, perineural disease, response rate just is nothing. It's a very surface two-dimensional measurement that does not reflect what's really happening to that patient. But the other thing I think about, like when I from other random part of my background here, is the fact that I was really struck in your book by the way that I think this is what you were trying to imply, that companies train us to believe in bioplausibility, that there is a process that happens. You can interrupt me if I'm being unfair.
No, you're right.
That you go to this conference and there's this nice booth set up, and you go in there and there are these pictures of I don't know, molecules moving around and something going, lock going into some kind of key, and then they show you like how that works, and then they talk to you about it, and maybe this drug even hasn't even been released yet, right? But you already love the story. You already saw the pictures, you already saw it. What happens. I mean, I think that's what you were implying in the book. And, that really speaks to me in terms of what you were saying about history, because as someone who spent a lot of time studying cultural history, this is historiography, right?
Yes.
This is the meta history of what happens, which is that we write history to explain stories to ourselves and to make other people believe that story. But when I started thinking about that, I got to be honest with you, Vinay, it kind of freaked me out. Like, I was like, whoa,
You're a player in history. I mean, I think you're accurate that is an accurate characterization of my views. I'll give you a good example. Recently, I was on Twitter and I saw an ad that came to me based on what I like to look at, which I guess is oncology, and it said, "Some numbers matter," and then it said 100% of multiple myeloma has BCMA, B cell maturation antigen. What BCMA drug is yet approved by the FDA? None yet.
But they're setting it up. They're setting it up. They're laying the ground.
They're laying the groundwork.
The bridge is being built.
The bridge is being built. And so now,
that's BCMA, scary, man. That's really scary. I'm just going to tell you.
That's how, I think, and if I'm perfectly honest, and, I know we have some people who run conferences, but, and this doesn't speak to Astro, but one of the, I think the major purpose of oncology, medical oncology conferences, or at least one of the ways in which it's been exploited. We all know the central gala, the displays that have carpets so plush you can twist your ankle in it. We all know that. And we all know that much of the content, I think, is meant to give us a favorable impression of products before they even launch. I'll give you one example. If I had you like lay on a psychiatry couch and I said words like vincristine, etoposide, cyclophosphamide, you're going to say, yuck. Oh, that's a toxic, horrible drug. Etoposide, oh, it gives me a creepy feeling. Then I say words like ibrutinib, venetoclax. I say words like that, and you're going to be, wow, that BTK, that's a really important target. BCL2, but what's the difference between tubulin, paclitaxel's target, and BCL2? They're very dirty targets. They're targets that do a lot of stuff in cells. But BCL2, that's targeted. That's exquisitely targeted. Tubulin, even though paclitaxel is the most potent inhibitor of tubulin, that's a dirty cytotoxic drug. So, I mean, all of the ways in which we even have this sort of visceral response to drugs is not based on an impartial consideration of the evidence. And, we published a number of papers looking at response rate by class of medications now. It's, I think, based on the fact that we are sort of constantly steeped in this environment where we are kind of sold new drugs before they come by the industry. I think that's part of what they do. They may not even be aware that they're doing it, but the same thing happened with the SSRI class of medications. Prior to SSRIs, companies spent tremendous money to get you to change your thinking around depression, that it was a chemical imbalance. And that was sort of paving the way for the launch of Prozac. But this is a tactic that's been used for a long time.
I mean, I hate to say it, but I'm going to tell you a little secret, and I hope you don't write your next book about it. The same
No one's
thing.
listening.
No one's listening. There's nobody.
No one's listening.
It started to concern me because I was like, is the same thing happening in radiation oncology? I can tell you right now, we have this, this new technology called Flash that's very fancy. super fast, flash.
Flash.
And, I love it. Everybody loves it. It's an incredibly amazing thing that, where you would treat patients to incredibly high doses of radiation with zero toxicity. I mean,
The Holy Grail.
Come on.
Sounds
Yeah,
great.
you would want that.
Sign me up. I don't even have
But the fact is that the companies are training us how to think about flash. So a lot of the education about flash is coming from the companies themselves. And I'll just say at the Red Journal, I was really proud that we published an editorial, which I can point people to, by Dr. Hendry, that really talks about flash in a different way, like the history of the science and what flash really is and is not. And it just, it's a very, very different perspective. So let me ask you something here. This is the hard question. How do you get outside of that system? How do you educate yourself about things that are going on? I mean, legitimately going on, right? Like, I can't not know about flash. Right. You can't not know about drugs. Like, that's not going to work. So how do you educate yourself within a system that is so steeped in teaching us these stories in a very structured way, right?
Yeah, that's a really great question. And
it's kind of like, getting outside the house, master's tools won't destroy the master's house. How do you get out of that?
Yeah. And I guess, I don't even pretend that I am fully out. I'm probably as ensconced as anyone else. I might just have a little piece of me out, and that's what led to a lot of work. But I think ways in which you kind of burst that bubble. I mean, one honest thing is, often it does take new eyes to burst a bubble. So sometimes it's the medical student on rounds with us asking a question like, well, why do you really do, where do you really do that? Or where is this response? Why is it 30%? The medical students got the confidence.
You go to the medical students. Wow. Okay. That is not what I was expecting you to say.
But sometimes it's the person with the freshest eyes.
The next medical student that rounds with me is going to be terrified.
Tell me something I don't know, medical student, and impress me. Impress me. This better be good.
Something completely outside the system.
So I think that's one. I'll give you a couple.
You're pointing to the idea of kind of unvarnished inquisitiveness,
yes,
right?
Unvarnished.
Yeah, or sort of uninfluenced.
And we forget, after many years of practice, we don't even see the things we are doing, the choices we're making in our practice. But when you were the first moment you were an intern, all of those choices were like visibly apparent to you. I think that's one. I think the second thing is, sometimes it's a litmus test to talk to people. I have some friends, some high school friends who, are in all sorts of different walks of life. And every once in a while, I tell them about the game changer drug at the national meeting. And one of my friends once asked me, what was the survival without the drug? And I said, it was a median survival of 6 months. I said, what was the survival with the drug? I said, 11 months. And I was like, that's great. That's almost a doubling of, and he's like, 11 months, get the hell out of here. Do better than that. He's like, what's wrong with you? You lost perspective?
I love the game changer part of your book. I mean, I was actually sort of, again, horrified and very disturbed that you had actually gone and investigated this on Twitter and found that that language was used on Twitter, game changer language, sort of meaning extraordinary language or superlative language, by people who had received more compensation. And that was really, it was eye-opening to me. I was kind of wondered as an editor, because I've been an editor my whole life. I'm always editing people for some reason. It's just this thing I do. And, game changer, I'll just tell you, game changer language really bothers me. It's always bugged me a lot. And I always wondered why. Now I know. Now I have a reason to suspect it. But yeah, so anyway, it's difficult, right? Because you also, sorry about that. You also have to, how can I put this?
Nobody likes game changers.
It's a marketplace of ideas though, Vinay.
Yes.
And the reality is that I can say this as someone who's steeped in the clinical trial world and has to justify, my choices and my decisions about things a lot. It's really hard to avoid that kind of language when you're trying to essentially to sell, right?
Oh, well, that's
and again, fun. I think this gets back to like, how do you get outside that system? Like, how do you advise people to, to work within that system in a way that is, that has integrity?
Yeah. I guess, just to clarify one point, we did work on Game Changer, but the specific thing you're talking about tweets is we did show that if you are paid by a drug company, you're more likely to tweet positively about that drug than you are about other drugs from other companies. And in fact, probably disproportionately about that drug. But in terms of this salesmanship, this is kind of baked in. This is, I think, one of the deepest, most corrosive problems in science, which is that you want scientists to be very dispassionate, analysts of the evidence. And dispassionate analysis would tell you that most putative drug compounds are going to fail. They're not going to come to market. But we incentivize grant writing. We incentivize money. We incentivize trials based on the person who can make the biggest pitch for their product. And I think that is a fundamental failure in the system, is that we make good scientists bullshit artists to some degree. We encourage them to upsell what they have, to oversell their results. And that is a very corrosive thing that I think it's very difficult. There have been some proposals like this PQRST model, some things like that. There have been different proposals to try to tackle this, but the fundamental, I think, failure is to recognize that the best scientist is not the one who has the discovery. That's often the most serendipitous scientist. The best scientist is the one who does the most honest work, knowing that some of us will succeed, but most of us will fail.
So let me ask you a question then. So if you had to redesign the nationally funded clinical trials system, how would you do that? I already know how you would redesign the FDA, but let's talk about the clinical trial system because I care more about that.
Okay. Well, I mean, the first thing is you're going to like it because, well, I mean, let's just put some numbers here. I mean, one reality is we currently spend at least $1 trillion on healthcare spending at the federal level in direct payments, Medicare and Medicaid. We spend even more in employer-based subsidies. So we're spending trillions of dollars as a society on healthcare. That's one fact. The next fact is how much of that money is being spent on things that we legitimately do not know if they make people better or worse the way we're using them. We just legitimately do not know. And the answer is there are different estimates in the literature, but it wouldn't be surprising if it's anything between 20 and 50% of things we just have no idea about. And what percent of expenditure is that? That could be as much as a third of federal expenditure. And there are a number of sort of annual papers that say what percent of healthcare is waste, and it's always so massive. What do we spend on clinical trials? And let's just talk about non-conflicted clinical trials. Let's just take cancer medicine. You know, we're spending $150 billion globally on cancer drugs. The NIH budget for NCI is something like $5-6 billion. And of that, only a sliver is for clinical trials and cooperative group studies. I think that ratio is broken. I mean, if you work for a company and you're saying, we have a $100 billion outlay each year, and I have no idea if this is helping our bottom line, it'll cost me $1 million to test whether or not this $100 billion expenditure is valuable. Should I do it? The company's going to say, yeah. And because if it turns out it doesn't help, we can save $100 billion. But we don't look at it that way in the federal system because obviously lobbyists and things of that nature. So I guess one thing I would do is I would crank up. You have to slowly and steadily crank up. You can't crank it up all at once. You got to ramp it up. Federal investment in clinical trials. You want clinical trials that are run by non-conflicted groups. You want the people who design the clinical trials, who think of the endpoints, to be academicians who are as non-conflicted as possible. And then you want a research agenda that prioritizes questions based on something called value of information, but that basically means things that are likely to make an impact, likely to save a lot of money, prevent a lot of toxicity. That's what we should prioritize versus things that are used less frequently and less likely to be toxic and things like that. And so I think with a really robust public effort, I mean, we could take a minute and just point out the fact that the pandemic has revealed nothing less than that the US, what was the greatest nation, has a failure to commit to public science. We failed to invest in public health, and we failed to invest in clinical trials. And we have to learn that dexamethasone is beneficial in COVID from the UK study, because we couldn't get it together to run that trial here, even though we have 8 to 10 times as many patients with COVID. I mean, I think that represents, structural failures in the US system. And so I guess I would crank up funding. I would try to make it as non-conflicted as possible. One of the problems with conflict and getting money from the industry in oncology is that people who do that have better careers. We've proved that in some papers. We need to reward junior faculty who stay non-conflicted. And maybe the reward is you get to design these clinical studies in this huge budget, this agenda. So anyway, those are just some ideas. I think like the epilogue of the book kind of gets at this space. But yeah, I think it's a really important question.
I do agree with you. I have to interject just one quick question. Any morning after thoughts on recovery? Any regrets on the tweets or the advocacy? Because the real publication's out now.
I was right. No, I mean, I guess
no regrets whatsoever.
No regret. I mean, I guess
did it all.
I own it. I pushed all my chips in.
You do say in the book you don't like preprints. You do say that.
Yeah, what do I say? I don't like medicine by press release.
Oh
which
yeah,
is
press
what,
release.
yeah,
Press
medicine
release.
by press, which is what it was. I guess I would say, I mean, I think I don't like medicine by press release because typically that means something like ribociclib just had a impressive response rate in prostate cancer or something like it. That's what it typically means. In this case, I think it's a unique example because one, the drug is pennies a day. It's not the company. No one has financial stake in the company. Two, they published the protocol. You could have read the protocol for a month before. The statistical analysis plan was on the internet. The press release shows you all of the endpoints, the interaction coefficient. And now that I read the preprint and the supplement, there was nothing there that really changes the conclusion. And I think that's a view that most people have shared. But the debate at the time was we shouldn't trust press releases. And I guess I think that that's another example of, we had just been burned. We had been burned by Surgisphere. We had been burned by, clearly faulty and fraudulent or likely fraudulent research. And so we were apprehensive, but I think we took it out in the wrong place. So I think recovery was a good study, and I applaud them.
Yeah, I'm going to go back to something you said. I do agree with you that the incentives in the clinical trial world, I'll use a different term. I think they're misaligned. And I do think that, I think we had talked about this earlier this week, just the fact that the dependence of university and medical systems on soft money. Yeah. And what that has done in terms of, like all my clinical trial friends basically sort of say like, well, you eat what you kill. Yeah. Not my rad onc friends, because they don't really know anything, about that world, but my medical oncology friends who taught me everything I know. And it's a very different set of incentives. And I do have friends in the UK, I'm familiar with their trial system. It does run a little bit differently. I won't say they have so much more money. I do think funding is part of the issue too. And I think they're facing, similar pressures as we do on a lesser scale. But the fact is that, it's hard to run publicly funded clinical trials because the universities require so much overhead at this point. It's so expensive. How do you close that gap? You can only close that gap by going to sponsors.
Yes.
Right. And that incentive, I agree with you. It's misaligned. And I just, I'm not sure how to solve that other than, we have to somehow unhook the addiction to soft money.
Yeah, I guess I would say some people might view this as a great success of right-leaning economical thinking, which was that a gradual disinvestment in federally and publicly funded science, coupled with Bayh-Dole that has incentivized universities to pursue patents and profitability, have really kind of changed this nature of public universities. Public universities are they're a fish tank. They're a fish tank for like startup ideas. Everybody wants to be a laboratory scientist who's going to have their spin-off startup company. That's what the university wants to invest a lot of money in. And we have become addicted to private funding. And insofar as we have that addiction, it builds this soft funding model. It builds all of these perverse incentives. And you're absolutely right that, who does many universities really want to hire in medical oncology? Do they want to hire somebody who's going to come and get grants from maybe NIH, maybe one PCORI grant, sorry, not NIH, let's be honest, a PCORI or HRQ grant and do kind of policy work? Or do they want to bring somebody on board who can bring in 20, 30 clinical trials with the industry? And these are clinical trials that are written in a way that they're going to pay $30,000, $35,000, $40,000 per head, and it's going to cost us $20,000 per head. So we're going to have a huge slush fund at the end of that. That's what we have created, the incentive that trialists, quote unquote trialists in medical oncology are often people who have good pharma connections and are willing to agree with the pharmaceutical company's plan. And we are incentivizing that. And that's the most recruited phenotype in the space. And it has been a policy success by some folks to move universities in this direction. And so the sciences, which were once, I think, incredibly critical of the private sector, have now become a feeder for the private sector.
Yeah, I don't know how to solve that other than advocacy. I do want to ask you about one other thing, which is that, going back to this idea of response rate, one of the things I've noticed in a lot of clinical trials is that, there's a lot of pressure, and I'll say this is even true within the national clinical trial system, to find short-term endpoints.
Oh, yes.
And I've often wondered about this. It's like, we've really shifted from, what I would say, I like to think that, radiation oncologists, like I said earlier, we love overall survival and we're still like advocates of overall survival, but very few people are anymore. You know,
That's
and
the
we're
only
going,
reason you guys invite me to talk.
I mean, we're going to shorter and shorter endpoints, right? And
Yeah.
response rate is the shortest endpoint of all. And
Oh,
why are we going to shorter endpoints? And one of the things I thought was really interesting in your book was the connection to patent life, sort of drug patent life. And I kind of didn't really, I started thinking about that a lot, just, you know, the timing of that, like how that actually works, which I'm not that familiar with, but it seems like, you know, it's almost like if the drug patent life for 40 years, we'd probably be able to have longer endpoints. But because the drug patent life is sort of set at this arbitrary, what is it, 20 years or something
it's
like
20
that?
years from the patent.
Yeah, I mean, you know, you kind of have to have this very accelerated timeline.
I mean, I guess I would say, I don't know, maybe I viewed it just slightly differently than that. I guess I would say the thing about all of these intermediary endpoints, from minimal residual disease to response rate to all path CR, is that one, they're easily measurable. Two, I'm not sure they save as much time as we think. So we did a study where we noticed that all these cancer drugs approved based on response rate, they're often approved based on response rate, the percent of people with 30% or more shrinkage, plus duration of response. So of those people who had shrinkage, we followed them 16, 18, 20 months to know the median duration of response was durable. And then we actually compared that against measuring survival in a randomized trial. And it turns out it's actually not as much of a time savings as you would think. It's something like it brings drugs to market 11 months faster on an 8-year time horizon. It's a paper we published in JAMA Internal Medicine. But what I think the benefit is not necessarily the time savings, but the fact that these surrogates are much more likely to be positive than survival, at least for drugs. That of 100 drugs that can improve a response rate, we don't know the right answer, but maybe only 1 out of every 5 or 2 out of every 5 actually make people live longer, live better. But the company's incentive, of course, is to get the approval and get the market share. So I think it's a product of success rate. It lowers failure rates. And I'm not necessarily convinced that it speeds time as much as people think because of the nuance of durability.
That's interesting. I mean, it probably has to do with the fundamentals of statistics itself, right?
Yes,
Because
yeah.
you need a large effect size. And
Yes.
so maybe you're not going to get a large enough effect size if you use survival as opposed to something like response rate.
Yeah, and the way around it, of course, is to pick the sickest patients, to pick people with high event rates, and then at least the same hazard ratio will give you an absolute faster time to result. So one of the things that we think—
Well, I'm going to stop you there because I don't think this is actually one of the things I was going to ask you about.
Okay.
Do you think the sickest patients are in clinical trials? I'm not sure.
No, they're not. Of course not. Yeah, no, I think that they're patently excluded from clinical trials. What I was saying that is if you had a system where you told drug manufacturers, look, you're going to have to improve overall survival if you want these drugs on the market. They're not going to do pertuzumab in the Cleopatra study in frontline metastatic cancer. They're going to go third line, and then they're going to try to move it forward because the third line trial is going to result faster. But that's a great question. And then maybe we'll turn to audience questions after this one. But I guess the answer to your question, of course, is that we know patients on clinical trials are 10 years younger than average US cancer patients. They have far fewer comorbidities. They're much more likely to be white. They're more likely to be men than average Americans. And so we have racial, gender, and age differences. And those very likely lead to differences in actual a sort of efficacy effectiveness gap. That there are drugs like sorafenib and HCC that have an efficacy, but do they have effectiveness? Do they actually make people live longer, live better in the real world? And I have my doubts for many of these drugs with narrow therapeutic windows.
Yeah, I mean, it's very interesting because if you actually look at the percentages of patients who could qualify according to those criteria, it's a small fraction. It's probably like a third of the actual patients with that condition could have qualified for that trial. And so it does raise, questions about generalizability, and diverse. I mean, in my mind, diversity as well, right? We're clearly leaving out, huge sectors of the population. The NCI has actually tried to correct that. They've corrected their language such that they no longer exclude people with second malignancy as much.
And HIV and another sort of correction.
They're encouraging HIV. I think, there's a movement now to try to include more elderly. But the fundamental, again, you're running up against this issue that, the companies want the best performers. And this actually is another thing that really irritates
and the trialists want to win.
Well, this is another thing you can trot out to please your radiation oncology friends is that it's very difficult to run clinical trials with radiation therapy because it's always consistently regarded as an element that would introduce more side effects.
Mm-hmm.
And no one wants their drug to be associated with, the radiation oncology side effects or,
That's
enhanced by that. And so it's actually very, very difficult to do meaningful trials in that space.
That's interesting. Let's have people type questions, and while they type, I'll just say a couple things on this topic. One, Lou Fehrenbacher, who used to work in Kaiser Vallejo, did a really nice study of consecutive patients in Kaiser and eligibility for 2 clinical trials. I think one was the Sandler carbopaclitaxel study, and he found, just as you say, 20% of people in Kaiser who come in the door would be eligible for these studies based on the strict inclusion criteria. And then I guess the next thing I would say is that there've been a number of efforts within FDA and outside of FDA to basically push for broader populations in trials. But I think they go about it the wrong way, because it's not a matter of the inclusion criteria. If you took away all those rules, you're still going to get the same patients on trials. You have to, I don't want to say punish, you have to enforce the population. What do I mean by that? If you do your clinical trial and people are 10 years younger than average and they don't reflect demographics of America and they don't have any comorbidities, I believe that we need to craft a new regulatory pathway. We'll give them accelerated drug approval, and we'll do a post-marketing commitment to prove that the drug works in broader population, and that'll be the study required for regular approval conversion. So I think that the FDA needs to sort of use the stick and not just the carrot in this space.
Right. I mean, I think one of the things you point out in the book is that accelerated approvals aren't followed up on, right?
Oh,
They're given generously, and then there's kind of no back end.
That's, I think, one of the great failures is that a couple of Government Accountability Office reports and a number of papers have shown that the post-marketing commitments that we do have for cancer drugs are often delayed, ignored, neglected, and sometimes they do a post-marketing commitment, but it's the exact same surrogate endpoint that led to drug approval. It's not survival or quality of life.
Yeah, I mean, here we're getting into like, what's what can we do about the FDA? And I'll just say I find this to be a really difficult struggle. I mean, part of my work is in lung cancer, and you've got to be aware that there's been some incredible advances in targeted therapy with lung cancer. I think you talk about that. You sort of talk about like, what if the patient's a miracle? How do you kind of psychologically and mentally process that? And it is true. I mean, we have put patients on, BRAF inhibition and seen amazing responses in lung cancer that we just never would have seen, right, with anything else. And it's a very difficult thing to sort of say, like, no, that's a rare outlier response that may or may not lead to survival in this particular context.
Yeah, that's a good question. I mean, I guess I would say that, I mean, I absolutely agree.
And I do like your idea about precision medicine. I mean, we've had this own discussion in my cancer center just about, the paucity of real results in terms of precision medicine, right? Like, you test all these people, very few of them actually end up being in the right place at the right time to qualify for a trial. Mm-hmm. And the endpoint is very small, but then there's this future potential, right? And are you kind of sort of missing out by shutting that down? I'm guessing we're not the only cancer center that's struggling with that, and the immense amount of expense involved, right?
Yeah.
You may have a more clear view on this.
I guess. Well, let me make a few points people putting in the comments, and I see Brian has popped up. So Fran is pointing out that patients with disabilities are often excluded from clinical trials. I think that's an excellent point. Anna is asking, what role does advocacy play in helping address the issue of pharmaceutical influence in clinical trials? I guess I would say, I think, part of the reason why I even tried to write the book was to sort of bring awareness to these issues. I think that, as physicians, that we're in the prime position to kind of push back. We may not be able to initially kind of change how the clinical trials are done, but at least we can change the dialogue at national meetings. I'm often, why is this podcast called Plenary Session? It's a bit of a joke because it's the exact opposite of the actual plenary session at ASCO. I'm not a cheerleader. I'm not going to cheerlead for all these drug products. I'm going to often pick things that I'm picking because I can rip them to bits. And I think that the more we kind of teach the skills of evidence-based medicine, the better we do at being critical and being sort of honest appraisers of evidence. Matt is asking, how well do you think oncologists trialists measure living better, and what can be done to improve quality of life measurement? That's a great question. We have a paper showing that, one of the fundamental failures is we only measure quality of life on drug till progression. And the moment people progress, we don't measure quality of life. So it's possible that drugs improve PFS, they improve quality of life initially, but it's followed by rapid progression, no improvement in OS, and deteriorated quality of life. We don't even know that. And so I think quality of life has to be measured in everybody, in every trial, and especially until the final thing that happens to cancer patients, which is often death. We have some more work on that coming out. Do you have any suggestions for how to support national clinical trials or IITs with radiation therapy that don't necessarily include substantial pharma or industry support? Now, that's a question for you, Dr. Yum.
Oh man. Yeah. I mean, of course there's there's grants, and you can try to sort of go, a more translational science pathway. But I mean, I think it gets down to the fundamental issue that Vinay and I are pointing out, which is that it's just so expensive to run trials, right? And so you can't really do that without support from somewhere, from your department, from your cancer center, from some public organization or advocacy organization that decides to fund you. And part of the issue here is this sort of, I don't know how we get out of this situation, this kind of dependence on soft funding.
Ramez is asking, great book, thank you. I'll pay you later for saying that. As a trainee, how do you remain critical while it is expected of you to just toe the line? That's a great question. And I guess you might not like my answer, which is wait for the moment. Wait for my general answer is wait till you're in attending, because you're still very vulnerable as a trainee. I mean, you don't need to speak out on every issue. It's good enough to read and keep a critical mind and follow things. And I think there's role to speak out. But you don't have to be the one who puts your neck on the chopping block. But wait for it. Wait till you're in attending. And then I think, in this profession, I think one of the things that's often unstated is, as a doctor in this country, and this is something I heard Malcolm Gladwell say when somebody asked him, why do you do all these stuff? And he was like, I've never been afraid to take risks. I live in American Canada. I'm always going to be fed and clothed. And as a doctor in this country, you're always going to be able to get a job. I mean, that's the reality. Some clinical job, you're always going to be able to put food on the table. So I think when you do become an attending, you should use that as license to take some risks and push back on things you disagree with. I think Dr. Kavanaugh has a question, but here's one. What should be the role of an institution's technology transfer office in promoting innovation while trying to protect cash on new drugs? Oh, well, I think that's a huge topic. And as we all know, often the universities part with very lucrative products for pittance. Just a fraction. I mean, people talk a lot about how much UCLA received for enzalutamide, which is in the billions. But the product, of course, is making potentially upward of $20 to $40 billion in life cycle sales. And so only a fraction goes to UCLA. CAR-T and Novartis and the licensing agreement, you can look into that. I think it's a big challenge. I don't think I know the answer to that. Let's see what Dr. Kavanaugh has to say. I see him.
Oh, well, I didn't want to interrupt Flo. You guys are awesome. First of all, two things. When I made up this stupid, ridiculous title to this thing, a bad pun, it's really terrible because I don't know what glass of wine I'd had that night. And then when I also said that subline about the interviewer and the interviewee, I didn't really think that was going to be serious either. But two, you have 100% turned the tables in a fantastic way. And Vinay, you're such a good sport. I really want to thank you for just rolling with it. I mean, I might have thought that she would have asked a few things about your book, which is terrific, and who knows what would have happened from there. No, could I just read? I just wanted to read my personal favorite line from the book.
Okay,
that's okay. And this is on page 188.
You're welcome to it.
Following along at home, and this is after he's talked about how there's perverse incentives all over the place, and he says our current regulatory system is the parent that spoils the child. We buy our sons and daughters a BMW or Ferrari for a D+ report card and then wonder why they don't work harder for A's. So I just want to celebrate that particular line, which I thought was spot on in a million thousand ways. There are more questions coming in, so I don't want to interrupt the flow. And if we really run out of things to talk about, Sue, you can talk about radiation therapy to the lung for COVID. We're not going to go there. No, not today. Not right now.
We don't want to talk about parachutes on this podcast.
Okay, go back to the questions. They're much better than what I have to say. Keep going.
Well, that's funny you raised that quote. Thank you for pointing it out. I think the poster child of that is Avastin. This is a drug that has life cycle sales of maybe $70 billion or more. And wherever it's been tested, it improves overall survival, usually on the order of about 1.5 months. In idealized patients in clinical trials, and maybe cervical cancer is on the high end at 3 months, and then a lot of cancers doesn't work at all. Here's a question from Sarah. Is proton therapy the sexy, expensive, targeted therapy of radiation oncology without high-level evidence to support it? Well, I hope Dr. Lee from Sloan Kettering is not on this podcast, but
wow, you know Dr. Lee, that's some insider knowledge.
I hope she's not on this pod. Well, I'm sure she's got better things to do.
I taped it for Vinay.
I'm gonna cut this clip out, but I guess I'd say you might not be wrong. Annie is asking, any comments on liquid biopsy testing?
I'm just gonna say the UK. Okay,
Yeah.
let's go back to the UK and CRUK, which, you know, there are a lot of problems with the NHS. Okay, I'll be the first one to say, like, I would have to hear more about the NHS and experience it more to go sign up for healthcare there and move to England. But that aside, and the issues with NHS and the fact that they're also privatizing because of their problems, the CRUK, which is the clinical trials arm within the UK, has actually set up very meaningful proton trials, real proton trials, true randomization, you know, no industry involvement, mostly supported by CRUK with philanthropy. And I actually believe that those trials that come out of the UK in a manner similar to recovery,
yes,
are going to be what we end up referring to, which is a hard pill to swallow.
Well, I'm already moving on to carbon, so I'll be past you by then.
Well, you got to go ask the Germans.
Any comments on TMB? So I think you guys will be look forward to. I think there'll be a pro-con commentary coming in Annals of Oncology. I'm going to be writing that the FDA's approval of TMB was misguided and erroneous, and then I believe Vivek Subbiah from MD Anderson and colleagues are going to be writing that it was a good approval. And so I think it's going to be a very interesting thing. I guess the only thing I would say is it's a drug approved on the basis of response rate. And there is sort of a relationship between response rate and TMB. If your TMB is less than 10, your response rate in that study was 6.7%. If you were 10 to 12 mutations per megabase, it was, I think, 12.5%. 12 to 20 was something, 12 to 20 was something like, I don't know, 20.5%. And then above 20, it's about 40% response rate. And so then the question I have, the philosophical question is, either you believe we need drugs that improve survival and quality of life, in which case this data is inadequate, or you believe if a drug has a response rate, well, then it's a doctor's duty to offer that to a patient. Maybe that's a potential for benefit. I don't know all the answers. But if you believe that a drug that has a response rate is something we ought to be offering all the time, then why did you restrict your approval to 10 and above? Because people below 10 have a 6.7% response rate, and that ain't zero. So I think that's going to be a philosophical challenge. Ian is asking, is there enough evidence for PBT? What is PBT? Further explore the magnitude of benefit.
Proton beam therapy.
Oh, proton beam therapy in pediatrics. Oh, well, that's going to really get me in trouble in this audience.
Not necessarily. Not necessarily.
Yeah, I think not necessarily.
I think you'd be surprised. No, I mean, really, I mean, I think, I mean, how can I say this? You know, one of the weaknesses / vulnerabilities of the radiation oncology community is that we do allow a lot of dissent, and there's a lot of mixed opinions in the community. And we critique ourselves the hardest. It's such a real pain to be a radiation oncologist, let me tell you.
Well, it's part of the reason I have such admiration for you is that you've long had a tradition of doing really good cooperative group studies. You're powered for overall survival, and you tolerate dissenting opinions, and that's why I'm giving the keynote. I fear for that system,
Vinay. I truly fear for that system. I'm being really honest and heartfelt right now. I fear for that system over the next 5 to 10 years, because the fact is that the public doesn't understand it. You know, they're talking right now about what is it, 9 or 11% cut to NCI or NIH next year. It's, you know, it's just constant. I mean, we just had a cut. So it's like ongoing issue.
COVID-19, even when you do everything wrong, things go bad anyway. That's going to be the book I'm writing next. Philippe is asking, in the book Malignant, I say imatinib for CML is a good drug, probably one of the few parachutes, any other drugs that are that good? I guess the answer is no. And just to put it in context, like, why is imatinib so good? You know, if you looked at, let's say, a hypothetical 55-year-old woman who was diagnosed with CML in 1974, her life expectancy with CML is about 3 to 4 years. But the 55-year-old woman who, if she didn't have CML, her life expectancy would be 22 years. And so that's a you know, an 18-year gap in life expectancy, years of life lost. That same woman in 2010, that gap is down to about 1.5 years in the Swedish data set. So imatinib is such a transformative drug that it closes a 20-years of life gap. Let's just pick another example. ALK rearranged non-small cell lung cancer. We got a bunch of good drugs. alectinib, crizotinib, brigatinib, lorlatinib, you name it. The same 55-year-old woman diagnosed with that cancer in 1975 probably would live, she probably wouldn't live the average of a lung cancer patient. She's probably a little bit longer, but maybe something like 20, 22 months, maybe a little bit less, maybe 15 months, but certainly not the average, because we know that mutation is often a favorably prognostic in and of itself. And what is that person living today? Maybe 5 years in the best clinical studies, if that's a trial eligible patient, but that still leaves 15 years of life lost, which is the tragedy we don't see. We see the 5 years full in the glass, and we miss the 85% empty glass. So I think that puts in perspective, and that's a really great drug. So most of the drugs we have are, I think substantively inferior to imatinib.
Hey, can I just mention one thing?
Yeah.
Only because I'm happy to go all night. I mean, it's fine. We can keep going.
Oh, well,
some people may consider it an hour as reaching an hour, and that's maybe an okay thing. Since we were doing all this kind of funky cross promotion things like this, I happened to see your Twitter recently that a certain Dr. Vishal Gewali, who I think is in the audience right now,
is he
going to be one of your upcoming guests. And
yes.
Vishal, if you feel like going on now and just saying anything you feel like, we would certainly always love to hear you. So I'm going to unmute you, or maybe Kaylee can unmute you. And if you're still there, if you want to say anything, we would love to hear.
And I'll introduce Vishal. I mean, I guess Vishal Gewali is, of course, assistant professor at Queen's University, a practicing medical oncologist, originally from Nepal, who is interested, I think, in regulatory policy as well as global oncology. He's coming on the podcast in the future to talk about what has become of Twitter. And I think it's going to be a fun, it's a discussion that he said is going to get him in trouble. So Vishal, it's great to hear from you.
No, it has been a pleasure being a part of this live recording of the podcast, and to be a fly on the wall in the radiation oncology room. So now for the first time, I'm getting a sense of what it feels like to be, a part of the radiation oncology team. And it feels like, we are a bunch of people who are really thoughtful and who care about evidence. And I guess that's a selection bias for this webinar, I would assume, but it really felt good to be a part of this. Thanks for inviting me.
I don't know, Vinay, if that's been on your agenda or if I missed a podcast or two, the global oncology theme, which is just a terrific topic. In your book, of course, did we mention that his book is Malignant? Yes. Okay. In your book, one thing we didn't get to here has to do with the fact that a lot of companies will, I'm going to go ahead and use the word exploit, developing countries for the purpose of using randomized studies against strawman comparators. That's a big part of this book, and I think it's one of the important points. And Vishal, I don't know, I'm not going to ask you that loaded question and give you 5 seconds to answer that. I'm sorry, but I know
but Brian,
that's a problem.
I have one thing that Vishal could speak about that will fit this audience in global oncology. Vishal gave a very excellent lecture at OHSU a couple of years ago, and one of the things he pointed out was the difference in radiotherapy's availability in Africa versus for a particular group of people, for some particular population in the Northern Hemisphere in the United States. Vishal, you want to tell him that?
Yeah, that was a news I saw in BBC, which compared the availability of radiotherapy machines for people in Africa versus cats and dogs in USA. And, there was, I can't remember the exact number, but there was one radiotherapy machine for the whole country with 30 to 50 million population. And then there were, almost 1,000 times higher per capita radiotherapy machine for cats and dogs in the USA. And of course, this is nothing against cats and dogs, but it's about how in certain parts of the world people do not have access to even life-saving interventions like radiotherapy and surgery. And the biggest paradox I notice in global oncology is that in these particular corners of the world where you don't have radiotherapy machine, sometimes patients are getting Avastin for the tumors, and they are paying out of pocket for that. So this is what I always mean by cancer ground shot before cancer moonshot. So before we get onto all these overhyped but low value interventions, the first priority should be to make sure that every people in the world will have access to things that we actually know to work. And the only thing that we need to do is to know how to implement those, rather than trying. Sometimes you see papers about how to implement precision oncology in Africa. And that reminds me of that news article comparing radiotherapy machines for cats and dogs in USA versus cancer patients in Africa. And that is really something that, it would not take uh, 5 or 10 moonshots to address that issue, right? It would take one sincere ground cert to address that issue, and we could do that.
Yeah, and I think that's such a heartbreaking statistic, and I think it really drives home the point of the challenges faced in global oncology. So thanks for sharing that, Vishal.
No, not at all. And I'd like to thank Astro for making the theme related to global oncology for this year, because that's quite amazing. And I guess this is the first big oncology meeting that has actually brought the issue of global oncology to the forefront. I just feel ashamed that none of our medical oncology communities ever thought of making global oncology as the forefront of their annual meeting. They do have some spin-off meetings, like second tier. We have second tier journals, right, for global oncology. And similarly, some of our communities do think of global oncology as a second tier research or second tier priority. And we do have some global oncology meetings for, like 100 or 200 participants who are who have a special interest in that. But to have the whole actual annual meeting with the theme of global oncology is what we need to make all of us aware about that need. So I really like to applaud the Astro community.
That's really terrific, and yeah, that's great that Astro is going to be talking about that important theme. And maybe I'll just take a couple more questions. Annie is asking, the AACR keynote talked about lives lost because of delayed mammography and colonoscopies. It was really hard not to think of arguments from your first book. Anything you want to remind us of? I was like, well, people are trying to get me in trouble. I guess I would say that, there are a couple things to note, of course, which is that cancer screening trials are primarily powered for cause-specific mortality or death from that cancer. That requires adjudication. It's often very difficult. And they're not typically powered for all-cause mortality or living longer, which is really the measure of lives lost. When it comes to colonoscopy, we actually don't have any randomized trials to date about looking at the full colon. We only have flexible sigmoidoscopy. But fortunately, flexible sigmoidoscopy in meta-analysis does now have a statistically significant overall mortality benefit. FOBT, unfortunately, does not. Mammography, however, unfortunately, in the Cochrane analysis and pooled analysis, does not have an all-cause mortality benefit. And we can talk about whether or not this will be the case. But I guess I've seen a number of publications that are going to look at whether or not delays from COVID are impacting medical care. And I just want to say, I would be very cautious about modeling. Current models are based on sort of very kind of confounded data sets. I think COVID is, for all the faults of COVID, one silver lining will be it'll be a natural experiment to kind of ask these questions rigorously. So we might be able to actually have an answer to that question.
I was actually going to ask you that question, Vinay. I didn't get to ask this question, but I had this already in my head. Has COVID destroyed the medical publishing industry? Like, what has happened? Like, I'm just going to say, as an editor, we've tried to not get crazy, but it's been really concerning. I mean, we're just seeing our sister journals left and right kind of flip out.
Yeah, I think we've seen a number of high-profile retractions. We've also seen fiascos in the preprint literature as well, including highly publicized preprints that some people criticized of. Hydroxychloroquine was pushed in probably one of the greatest public health debacles of the last 25 years, leading to shortages and things of that nature. And rhetoric has been skewed. I think it's going to be sort of a huge thing to unpack.
Yeah. My concern is, like, what kind of damage have we done sort of semi-permanently here? Like, what have we done to the status of peer review? What have we done to the credibility of journals in general? I mean, there are people now who say to me, well, what's the difference? Let's just preprint. Mm-hmm. And, I just wonder, like, after COVID's over, are we gonna all wake up, not drunk anymore and figure that back out? Or, have we enacted some kind of permanent change and dissolving of that scientific process, with the expectation that everything should be published tomorrow without rigorous review, and we just throw hypotheses out there and it's a marketplace of ideas. It actually keeps me up.
I agree with you. I guess I'm a little, I actually do wonder, is it the case that COVID is worse, or is it the case that COVID is just what happens in science on a compressed time frame? Is it just what happens in 20 years of science compressed in 6 months? And I guess I'll be curious to know. I think we'll do some empirical studies to kind of tease those two hypotheses apart. But I think you're absolutely right. I think a lot of us have been frustrated by by all the fiascos in science. Anyway, this podcast is brought to you by Surgisphere. Surgisphere, the best data this side. No. Okay, well, just
good try. Good try. They're out of money.
Just do two last questions.
They're handling all the lawsuits right now. They can't pay us.
I wonder if they are, what they're doing. Their website is down temporarily, of course. Sabine is asking, has medical Twitter improved the criticisms of trial results resign, or is everyone a COVID expert now? How can Twitter be used as a platform? I think Twitter is showing that, that there are people out there who I think are just spot on, thoughtful commenters on clinical trials. I've discovered people I didn't even know existed. I think a huge group of clinical trial statisticians have come forward, and they are some of the most thoughtful commenters on trials. And so you can go on Twitter and you can definitely find some pearls every day. But unfortunately, you're going to find a lot of garbage too. And a lot of people who even are have titles that would make one think that they are knowledgeable and experts have proven to be deficient. And so I think Twitter shows what everyone what everyone's really capable of when it comes to clinical trial appraisal. And Anna is asking, in the book you write, doctors on Twitter are more financially conflicted than the average doctor. That's true. Should we state our disclosures or lack thereof, as Dr. Gewali has in our Twitter bios? Yeah, I guess I would say when we did this work on Twitter, maybe this will be the last question, then we'll take closing comments from the host. When we did this work on Twitter, one of the things a reporter called me up was to say that, you've shown conflict is rampant among oncologists on Twitter. There's a link between conflict and, promoting certain drug products. How do we handle this? And they say, I don't know if you know this, but celebrities have this rule that if they are being paid by Vitamin Water and they're tweeting photos of them showing Vitamin Water, they'll put #sponsored. And I thought to myself that, isn't it ironic when doctors have to take ethics lessons from celebrities? And so I do think that, we probably should disclose in our Twitter bios our conflicts. Maybe actually in the book, I argue that, we should also try to minimize even having them in the first place. And then we certainly maybe should be cognizant of the fact that, if you're on the payroll for AstraZeneca, maybe you shouldn't be talking about Profound. So let's go through, we're going to give the last word to Dr. Yam, and then maybe we'll let Brian take the next to last word, and then we'll close. But I think this was a terrific session, and I really appreciate you all putting it together and doing it. And thanks so much for having me.
So thank you. I'll take the second last word. Sue, you can close it up. And we are looking forward, because we do have the understanding that you're going to talk more about Twitter on your next plenary session. So looking forward to that. And thank you both for fantastic conversation. We could go on for hours, but we can't. So we do have to call it a close. But Sue, you can send us on home from here.
Oh gosh, what to say? That was like 5 cups of coffee at 7 p.m. I'm sort of amazed, Vinay, with how much you contribute so rapidly and so prolifically to the conversation. And this has just, I think, been a very small example of that. And just really want to thank you for working with me.
Well, thank you. And I'm going to have to bring you on the podcast and flip the tables on you and
oh, man.
And go through your whole story.
I'm in for it.
And then the last person we have to thank is we have to thank Kaylee for putting all this together and getting it.
Kaylee and Brian, amazing job.
Right.
And very, very interesting and complicated format, I must say. Give you kudos on that.
And thanks to the participants. And I think that's a wrap. So stay tuned for the next plenary session. Thank you all for participating.